Hello, and welcome back to our next section of Chapter 1. This is dealing with fermentation. And the big question that triggered this kind of field of research was what causes fermentation? Now, we're going to be dealing with the time frame we're moving along. So we did like 1600s-ish for Leeuwenhoek and Reddy, 1700s-ish for Spallanzani and Needham and Pasteur, and now we're kind of in the beginning of the 1800s. So we're going to talk about Pasteur again. And so the big question of the day, people had been making wine for centuries, right? It wasn't the understanding of microbiology and fermentation that people knew how to make wine. They just squished the grapes and let it sit, and then you got this alcohol. But when you have these big productions like vineyards and you bottle all your wine and you let it sit for a while, and then you have to ship it, maybe if you are a winemaker in France, you ship it to England or you ship it to Italy or whatever. Some of those bottles, by the time they got to their destination, were spoiled. They were spoiled by what's called acetic acid, which is like vinegar. So if you could imagine getting your bottle of wine and you open it up and you're like, ugh, this doesn't taste like wine, it tastes like vinegar. So the driving force behind this understanding of fermentation was people didn't want their wine to taste gross. So Pasteur kind of set out to try to answer that question. So by this time, a pretty good understanding of microbes was building. The ability to see microbes under the microscope. We had good technology, better microscopes, way better than Leeuwenhoek. So now we're a couple hundred years later after Leeuwenhoek. So here were Pasteur's experiments, and he had 4 hypotheses that he was trying to determine what caused fermentation, and with that, how can we prevent the spoilage of the wine? We want to keep the alcohol in there. We want to not have the acid in there. So here's his observation, kind of the step 1 observation is you take some grape juice and you smush it, or grapes. You smush it, you get your grape juice, there's sugars in there, and you let it ferment. So they knew how to do that. What they hadn't done before up to this point is, slap some of that stuff under the microscope, and what do we see? So he saw these little guys here, which are the yeast. Okay, they're a little bit bigger. They kind of come in these two pairs. And then he saw the tiny little guys here, which are bacteria. So in this fermenting grape juice, he saw 2 critters. He saw some yeast and he saw some bacteria, but he didn't know which one did what or if any of them was even important in the process of fermentation. So he kind of went back to what he was just working on with spontaneous generation. And so he took his grape juice and he boiled it. He killed all those things, the yeast and the bacteria, whatever he saw. So he just took the grape juice, killed everything, closed it up, spontaneous fermentation, like spontaneous generation, no fermentation takes place. So we can reject hypothesis 1. You don't make wine by just letting the grape juice sit after you've killed everything. Excuse me. His next experiment was air. Just the exposure to air is going to ferment the grape juice. So again, he takes his flask, a swan-necked flask this time. He heats it up, right? Kills all the buggers inside, lets it sit, no fermentation. We can reject hypothesis 2. So a sealed flask does not cause fermentation. An open flask to the air does not cause fermentation. So the yeast and bacteria have to do something, because in those first 2 hypotheses, he killed them and did not add any more of those organisms back. In his third, he isolated some of the bacteria and isolated the yeast. So now he's got a little culture, he's got a little tube of bacteria, and he's got a little tube of yeast. So in his third hypothesis, he took the grape juice, he boiled the grape juice, he killed everything in there, but then he added, he inoculated with bacteria, sealed the flask, not perfectly sealed, so we're not like melting it shut. So he sealed it and he got acid, not alcohol. So we don't necessarily reject the hypothesis, but we need to modify the hypothesis in that bacteria ferment, they change the sugar into something else, but that something else is acid. So bacteria ferment sugars to acid, right? So that's our modified hypothesis for number 3. And then 4, he did the same thing. He took his juice, he boiled it, he killed all the microbes, but in this flask, he inoculated with the yeast. He let the yeast sit for a couple days or weeks or however long it takes to ferment. And then here is our alcohol. So he accepts hypothesis 4. Yay! We now know, thanks to Pasteur, that it is the yeast that ferment sugar into alcohol and bacteria ferment sugar into acids. So you may have learned different fermentation pathways in your general biology course. This is kind of the guy who discovered that, which is very cool. So what is Pasteur's connection to our understanding of pasteurization? Is that when we pasteurize something, we kill off the spoilage microbes. We don't sterilize it. So when we drink pasteurized grape juice or apple juice or milk, we're not drinking sterile drinks, but we heat it up just enough to kill off the bad bacteria, the spoilage microbes, which then makes it safe, or any pathogenic microbes. So in the winemaking, what they would do is they'd get their juice, they would sterilize it to kill the bacteria, and then inoculate it with the yeast. And then the yeast would have this nice clean grape juice without any bacteria, and then all you would get would be alcohol instead of the acid that would spoil the wine. So that's a big contribution to Pasteur. So he not only defeated the idea of spontaneous generation, but he explained fermentation and how bacteria and yeast ferment sugars in a slightly different pathway and get different products. And then last contributor to this idea of fermentation, a guy named Edward Buchner. He 1897, so now we're towards the end of the 1800s. So a couple of hundred more years have gone by since Leeuwenhoek. So Pasteur did his experiments with living organisms, the living yeast and the living bacteria in the grape juice. What Buchner did was he's like, well, do you have to have a living cell to do that, to get fermentation? Does it have to be a living cell? So what he did was he took some yeast cells, because we know that yeast ferment into alcohol thanks to Pasteur's experiments. He ground them down, kind of filtrated out, so it basically broke down the cells, broke them open, just got the insides of the cell, the cytoplasm, and any chemicals that were in the cytoplasm. Inoculated some sterile sugar juice, sugar cane juice, right? So it's got sugars. Inoculated that with this extract of the yeast cells, right? So just the enzymes, that's the extract, and you get fermentation. So yay. But then to control, he sterilized or boiled the sugarcane juice. No filtrate is added. No fermentation occurs. So he's like, okay, Pasteur, I give you credit. You figured out that's the yeast and the bacteria that cause fermentation. But he took it one step further. It's not necessarily the living organisms that do the fermentation, but the enzymes that are within the living organisms that do the fermentation. So Buchner's contribution is it's this enzymatic pathway, this enzymatic breakdown of the sugar that converts it into the alcohol or the acid. Okay, so that's the end of our discussion on fermentation. Okay, see you next time.